We know that heat acclimation is critical for competing in hot weather and might even help improve performance in cooler temperatures. But could heat training actually reduce your long-term aerobic fitness gains? In today's episode, we'll explore a study of 3 weeks training in either neutral or hot conditions and see whether the two groups improve equally in overall aerobic fitness or key cellular responses typically found after a bout of exercise. Heat adaptation, done through training at relatively low to moderate intensities in hot climates, is important for athletes getting ready to compete in hot environments. Doing so reduces heart rate and core temperature, increases sweating rate and evaporative cooling, and reduces thermal discomfort. Some argue that even if you are competing in moderate temperatures, being heat adapted can provide an advantage by increasing your aerobic capacity and maybe even the amount of hemoglobin, the molecule responsible for carrying oxygen in the blood. The challenge for athletes comes in finding time for a heat camp in an already busy training and competition schedule. Surprisingly, despite more than half a century of heat adaptation research, the question of whether a heat camp actually impairs your aerobic training has not really been addressed until recently. In a 2021 study, Slivka had untrained young males trained for 3 weeks. One group did so in neutral conditions of 20 degrees Celsius and 40% relative humidity. A second group trained in hot conditions of 33 degrees Celsius and 40% relative humidity. 14 one-hour steady cycling sessions were done, with the participants asked to ride at a rating of perceived exertion of 15 or hard. Basing effort on perceived effort automatically allows the workload to increase as the participants became more fit or heat adapted. Before and after the 3 weeks, a test ride of 60 minutes at a constant effort was done, and muscle samples were taken before and 4 hours after the ride. One key measure they looked at was PGC1 alpha, a gene that is turned on after training and that may be critical in building mitochondria, the aerobic powerhouse of cells. What happened with the two groups before and after the 3 weeks? On the left, we see the results of VO2 max, or the maximal oxygen that the body could use. Both the neutral and hot groups had similar VO2 max before training at about 38 milliliters per kilogram per minute. What was interesting was that VO2 max increased by about 10% in the neutral group, about what we would expect in this untrained group and with this type of training. However, the hot group did not increase their VO2 max after 3 weeks, suggesting that the response to training was blunted by training in the heat. On the right, we see the response of PGC1 alpha. Before the 3 weeks of training, the neutral group had a 6.6 times increase in PGC1 alpha after the 60 minutes of exercise, much higher than the 4.6 times increase in the hot group. No difference between groups was found after the 3 weeks, with about a 2.4 times increase in PGC1 alpha. It's possible that this lower initial response may mean less building of mitochondria, which might explain the lower aerobic fitness after 3 weeks in the hot group. This study is really intriguing, being one of the few to see whether heat training can blunt aerobic training, and we really need more follow-up work. For example, would we see the same responses in elite athletes compared to the untrained participants in this study? But if true, this has serious implications for athletes and coaches building a training schedule. It suggests that, while heat training may be an important tool, it is not possible to have a heat training camp that optimizes both heat adaptation and training itself. Instead, we need to recognize that training itself will take a back seat during these heat camps, and that needs to be made up elsewhere in the schedule. Hopefully this video fires up your interest into the fascinating world of environmental physiology. I'm Professor Stephen Chung, and I run the Environmental Ergonomics Lab in the Department of Kinesiology at Brock University. We post new videos on different topics in environmental physiology every Wednesday. Thanks for watching. We'll see you next time.